Hello and welcome to our session dealing with mutations and the process of DNA repair. So, what did we learn last week? Last week, we had a closer look at two processes by which the cells are dividing. We learned something about mitosis and meiosis. So, what is the difference? Mitosis occurs whenever more cells are needed. For example, after injury, or in disease, stem cells are getting reactivated and divide to repair the lesion. Mitosis always produces two new cells that are identical to each other and also to the parent cell. Mitosis, together with cell growth, is called the cell cycle. In contrast, meiosis is a different kind of cell division. So again, what we need to keep in mind here, that a human body cell contains 46 chromosomes that are arranged in 23 pairs. In sperm or egg cells, only half of those chromosomes is present. And the result of meiosis is actually production of those cells. Therefore, meiosis is sometimes referred to as reduction division. In meiosis, 4 daughter cells are produced in contrast to mitosis, and each daughter cell has only half of the chromosomes of the parent cell. Also, in meiosis, 2 sets of cell division are involved, and this allows production of a new diploid cell after fusion of a sperm cell and an egg. So, what cell types are dividing via mitotic cell division? There are actually quite few cell types that divide fairly quickly in our body. And the first example of such cells are actually hematopoietic stem cells or stem cells of the blood. They produce blood quite frequently. Another example of dividing cells in our body are hair cells. Also, fingernail tends to grow, or our liver cells that have a quite quick turnover. And this is actually also the problem in cancer treatment, because we can't specifically target cancer cells in cell division. And this results in hair loss and other side effects of cancer treatment. So, what we are going to be looking at today, and what are the learning outcomes? After watching 3 videos today, you should be able to explain the term mutation. You should be able to explain the differences between genome mutations, gene mutations, somatic mutations, and germline mutations. You should also be able to name an example of a mutation having a positive effect on a carrier and give examples for disorders resulting from chromosome and gene mutation. You should also know the 5 types of chromosome mutations and explain different types of gene mutations. You should know common mutagens and also appreciate that genetic diseases cannot be cured by conventional pharmaceutical interventions. So, what are mutations and how does the definition of the mutation look like? If you go on the web and look it up on Wikipedia, then you will find something like the definition provided here. A mutation can be defined as a permanent change in the genetic material of an organism. So, what are the effects of such permanent changes in our genetic information? First of all, mutations can be neutral. They can have neither positive nor negative consequences. This type of mutation is neither beneficial nor detrimental for the ability of an organism to survive and also to reproduce. And a very good example of such neutral mutations is the eye color. All of us have different eye colors, but usually those different colors do not affect our ability to reproduce. Another example of neutral mutations is the form of the earlobes. Some of us have attached lobes, some others do have free lobes. However, also here, this type of mutation does not have any consequences on the ability to survive or to reproduce. Finally, hair color is another example of a neutral mutation. Neutral mutations lead to polygenic phenotypic characters, and this is actually something you can see on daily basis. We have different forms of earlobes, hair color, eye color, and those mutations usually don't have any consequences on the carrier. Rarely, mutations can also have positive consequences, and they increase an organism the chances of an organism to survive. In reality, positive mutations are also the driving force behind evolution. A quite recent example of positive mutation has been discovered by researchers working on the HIV virus. Here, biologists at the University of Liverpool have discovered that a certain mutation in the gene, which is important for the immune system, makes the carriers immune from infections with HIV. And this mutation is a mutation in the so-called CCR5 delta 32 gene. So why is this mutation important? CCR2 is actually a receptor on the surface of white blood cells. And as you can see here, HIV is using this receptor to dock to those immune cells and to infect them. However, if there is a mutation in the CCR5, or if CCR5 is absent, HIV is not longer able to dock to the cells and to infect the patients. And this is actually the reason why the carriers of this mutation are immune to HIV. Unfortunately, in quite few cases, the outcome of a mutation can be negative. And this is the case in many genetic disorders. Genetic disorders that are caused by genetic mutations can be caused by genome mutations and gene mutations. And an example of genome mutation we already had a look at is aneuploidy, which changes the chromosomal configuration in a cell. As we have heard before, an example of genome mutation is trisomy 21 or Down syndrome, where 3 copies of the chromosome 21 are present. Another type of genome mutations are chromosomal mutations or abnormalities, where parts of the chromosomes are rearranged. For example, where part of an unrelated chromosome binds to an unrelated chromosome, and if this mutation is inherited, we can end up with a genome mutation and genetic disorder. In contrast to those genome-wide mutations, there is also another type of mutation which can result in disorders, and those mutations are referred to as gene mutations. Gene mutations can be single gene mutations, which is actually a small number of genetic disorders. An example of such disorders caused by single gene mutations are sickle cell anemia, cystic fibrosis, epidermolysis bullosa, or Tay-Sachs disease. Unfortunately, many genetic disorders are multifactorial. Those disorders are caused by mutations of multiple genes, and in reality those disorders are much harder to treat using gene therapy, as we will hear in our dedicated lecture on this type of modern therapy. Most genome mutations are actually caused by problems in the meiosis. More specifically, they can be a result of a non-disjunction of chromosomes in different phases of meiosis, leading to a change of the chromosomal configuration. In this case, if a non-disjunction happens, we end up with aneuploidies, where the configuration is different because we have 3 copies of a chromosome or just 1, depending on which sex cell is fused with a healthy sex cell. In addition, genome mutations can occur during meiosis as a result of faulty crossing over. Crossing over is a normal process during meiosis which ensures actually higher genetic diversity, because if related chromosomes are adjacent to each other, similar or homolog parts of related chromosomes can be exchanged. And this is a perfectly healthy process which takes place during fertilization. However, or technically this occurs prior to fertilization during meiosis, so I have to correct myself to be entirely correct. So, however, this exchange can also happen between unrelated chromosomes if the area on the chromosomes is long enough and similar enough. As a result, chromosomal abnormalities can occur or deletions can take place. So we can have deleted parts of the chromosomes leading to a shorter chromosome which lacks certain genes. We can also have duplications or amplifications which lead to multiple copies of a chromosomal region, increasing the number of the genes, and the outcome of such mutation can be actually quite serious, because you can imagine if the chromosomal region is coding for genes that regulate cell proliferation, we could end up with cells that divide in a very uncontrolled manner. Also, we could have insertion of unrelated chromosomal beads into other chromosomes. So we have addition of material from one chromosome to a non-homologous chromosome. Finally, another type of genome mutation resulting from faulty crossing over can be an inversion, where the orientation of the chromosomal signal is reverted. And obviously, because the orientation is reversed, transcription of those chromosomal region is gonna lead to faulty proteins or no protein production at all. Last type of genome mutation that can occur as a result of faulty crossing over are translocation, where genetic material can be interchanged between two unrelated chromosomes. So let us have a look at few typical disorders resulting from non-disjunction first. Again, we have this example quite few types, but this is a very classic example, so let us have a look at Down syndrome once again. Again, frequency of Down syndrome is actually fairly high. As you can see here, nearly 800 babies are born with this condition in England and Wales alone. And this frequency increases significantly with the age of the mother. The result of the Down syndrome at the site of symptoms is reduced muscle tone, leading to hypotonia. The eyes of the patient slant upwards and outwards. Also, Down syndrome is often associated with a fairly small mouth with a protruding tongue, flat back of the head, and below average weight and length at birth. And from the chromosomal configuration point of view, this is caused by the presence of an additional copy of the chromosome 21, as you can see here fairly nicely at the karyotype of a carrier. Another example of the trisomy is a trisomy 18, also referred to as Edwards syndrome. This type of genome mutation is slightly less frequent. However, it occurs in 1 in 3,000 births. Here, the syndrome is a result of the presence of an additional copy of chromosome 18 in the carrier. Importantly, this is also a fairly interesting syndrome, because this additional copy of the chromosome is not present in all of the cells of the patient. Here we are dealing with the so-called mosaic phenotype. Result of the presence of this additional chromosome number 80 is fairly low birth weight, along with number of other serious medical problems. Of those that survive to birth, around half will die within 2 weeks, and only around 1 in every 5 will live at least 3 months. If the carriers survive, the result is usually presence of multiple malformation, especially of the heart and kidneys, clenched hands, and overlapping fingers, as you can see here at the patient shown on this slide. Trisomy 13 is also an aneuploidy example. Here the chromosome number 30 is present as 3 copies, and the frequency of this genetic disorder is 1 in 16,000 births. More than 80% of infants with this syndrome die within first year of life. The symptoms here are again multiple malformation, incomplete lobation of the brain, very small or poorly developed eyes, cleft lip and cleft palate, congenital heart disease, and hypotonia. In contrast to trisomies, also the lack of certain chromosomes can result in serious genetic disorders, and this is the case in Turner syndrome, which is also referred to as monosomy X. The frequency of this syndrome is 1 in 5,000 females, and 99% are lost spontaneously in pregnancy. If the carriers are born, this results in short stature, the ovaries involute before birth. Again, here we do have congenital heart disease, puffy feet, and redundant skin at the back of the neck. And as you can see here, the second sex chromosome is lacking. Another example of monosomy resulting in genetic disorders is monosomy 18p. The frequency of the syndrome is 1 in every 40,000 births, and the result is short stature, variable degrees on mental retardation, and malfunctions of several organs. I hope I convince you that genome mutations can actually result in several quite serious genetic disorders, and in the next session we are going to be looking in closer detail at mutations at gene level. Thank you very much for your attention.